Hello everyone, welcome to our channel. I am Shrijita Ghosh. I am your tutor for today's lecture. I hope it will be very helpful for the aspirants. So let's start. The study of microbial growth. Growth takes place on two levels. Cell synthesizes new cell components and increases in size. The number of cells in the population increases. So the basis of population growth is binary fission. Now let's see. This is binary fission which is occurring. We have a young cell at the early phases of cycle. It has got cell wall, it has cell membrane. This is chromosome 1 and these are ribosomes. Now, a parent cell prepares for cell division by enlarging its cell wall, cell membrane and overall volume. Midway in the cell, the wall develops notches that will eventually form the transverse septum and the duplicated chromosome becomes affixed to a special membrane site. The septum wall grows inward and the chromosomes are pulled towards opposite cell ends as the membrane enlarges. Other cytoplasmic components are distributed randomly to the two developing cells. The septum is synthesized completely through the cell center and the cell membrane patches itself so that there are two separate cell chambers. At this point, the daughter cells are divided. Some species will separate completely as shown here, while others will remain attached, forming chains or doublets for example. The rate of population growth. Generation or doubling time, the time required for a complete fission cycle. Each new fission cycle or generation increases the population by a factor of 2. As long as the environment is favorable, the doubling effect continues at a constant rate. The length of the generation time, a measure of the growth rate of an organism. Average generation time, 30 to 60 minutes under optimum conditions, can be as short as 10 to 12 minutes. This growth pattern is termed as exponential. These are the basic nutrients for growth which are required. ATP is required for cellular processes. Carbon is very much necessary for the production of any macromolecules like proteins, lipids and carbohydrates. Oxygen for metabolism. Nitrogen for amino acid synthesis. Sulfur for vitamins, amino acids, structural stability of proteins. These are the elements which are required by microorganisms generally. The population growth curve. A population of bacteria does not maintain its potential growth rate and double endlessly. A population displays a predictable pattern called a growth curve. The method to observe the population growth pattern: place a tiny number of cells in a sterile liquid medium. Incubate this culture over a period of several hours, sampling the broth at regular intervals during incubation, plating each sample onto solid media, counting the number of colonies present after incubation. Stages in the normal growth curve. Data from an entire growth period typically produce a curve with a series of phases: lag phase, exponential growth phase, stationary growth phase, rapidly declining phase, and death phase. Lag phase. Relatively flat period. Newly inoculated cells require a period of adjustment, enlargement, and synthesis. The cells are not yet multiplying at their maximum rate. The population of cells is so sparse that the sampling misses them. Length of lag period varies from one population to another. Exponential growth phase or logarithmic or log phase. When the growth curve increases geometrically, cells reach the maximum rate of cell division. Will continue as long as cells have adequate nutrients and the environment is favorable. The number of cells growing greatly outnumber the number of cells dying. Stationary growth phase. The population enters a survival mode in which cells stop growing or grow slowly. The rate of cell inhibition or death balances out the rate of multiplication. depleted nutrients and oxygen, excretion of organic acids and other biochemical pollutants into the growth medium. The number of cells growing will equal the amount of cells dying. Endospores begin to form in this phase. Rapidly declining phase. The curve dips downward. Cells begin to die at an exponential rate. The amount of cells dying out numbers the amount of cells growing. The dead cells become nutrients for the growing cells. Death phase. The curve continues to dip downward. Most cellular activity stops. Endospores are formed and released from the parent cells. Basic phases of growth. Lag phase, new growth medium, period of delay while cells prepare to divide, log phase or exponential phase. Cellular reproduction most active during this period, generation time reaches a constant minimum. Stationary phase, state of equilibrium when number of cell deaths equals the number of cell divisions. Rapidly declining phase, cells die logarithmically, endospores are formed. Death phase, number of deaths exceeds the number of new cells. Potential importance of the growth curve. It implications in microbial control, infection, food microbiology and culture technology. Growth patterns in microorganisms can account for the stages of infection. Understanding the stages of cell growth is crucial for working with cultures. In some applications, closed batch culturing is inefficient and instead must use a chemostat or continuous culture system. Graphing bacterial growth. The data from growing bacterial populations are graphed by plotting the number of cells as a function of time. If plotted logarithmically, it will give a straight line. If plotted arithmetically, a constantly curved slope will get. To calculate the size of a population over time, NF equals to Ni into 2 to the power G. NF is the total number of cells in the population at some point in the growth phase. Ni is the starting number. G denotes a generation number. This is the population equation. Thank you for watching our video. If you like the video, please hit the like button and subscribe to our channel and share with your friends. And once again, thank you so much.